I am going to also start sharing a screen. So we're going to go through introduction to water monitoring to start off. And so feel free as I'm going along here to just unmute, or you can send a chat and hopefully I'll see the chat and answer any questions that you have. So first thing that we're going to cover today is just kind of a general introduction of water monitoring, and then we'll get into the specifics of water monitoring for bacteriological contamination. So basically around here, we once upon a time had lots of tons of salmon. They've been somewhat reduced now, which obviously has had an impact on other species like orca. Our creeks are really important because they serve as kind of nurseries for the salmon that would go out and feed the orcas. And I got involved personally because I live on a creek called Little Swamp Creek in Kenmore, and I don't often see large salmon in my creek, but I have once upon a time seen them, and this is a picture of a large one that we found that came up our creek. I've talked to people who lived on the creek in my yard long ago, and they used to see quite a few of them. Sometimes, generally the water quality is good, but sometimes it's not so good. So this is an instance of water quality that I saw last fall in our creek. And you can kind of see, this is a chart of what's called turbidity. Turbidity is a measure of cloudiness in water. And so normally around here, the way that we measure turbidity is in units called either NTUs, and we'll get into that in the water chemistry class. There's different units for measuring, but essentially around here we have very little turbidity, close to zero. But we had on that particular data, it was up at about 50, which is way out of norm. And the other thing that can happen is when you have high turbidity in a creek, that can also cause high bacteria levels. And so you can see, when we'll talk later about bacteria, relatively safe is under 200 by the units that we measure bacteria in. Washington state standards under 100. It was over 2,700 on that particular date. And I had another instance of a similar thing. So that's my connection, is that I live on a creek, and I've kind of talked about you guys and sort of what's your connection. So basically by learning about your stream or streams that you're concerned about, sharing that information with others and letting people know when you find a problem, you can help make things better for our local creeks, which is ultimately going to benefit salmon, orcas, the whole ecosystem. So a question I have would be, why do you think it might be important to monitor water? Anybody want to chime in an idea on that one?
Well, one of the reasons I'm interested in it is because I was talking to you earlier about this, but establishing a baseline for any future measurements we might make. One of the problems that I've been having with just trying to figure out what kind of climate action plan works for Lake Forest Park, for instance, is we don't really have any historical data. So it's really hard to say what our targets ought to be. And so by at least doing this work now, for future generations, we might be able to say, okay, in 2019-20, it was like this. And, you can base your, plans off of something like that.
Yeah, that's one really great reason, is establishing some baseline data. And some of the just really simple data that we collect on a baseline is air and water temperature at the sites that we monitor. So when you've done that for a long period of time, you can kind of see some trends.
I think it's also important that as volunteers, we can help advocate if we see issues. We can only advocate if we have good data. So I really like the protocols that you all do. And I know the two times I went out with my fellow monitors, it was a good engagement opportunity for those other folks that live in our watersheds. So two other great reasons, I think, to do it, and to do it as a volunteer is powerful in your own community.
For sure. Sarah, do you have any thoughts?
I mean, I'm definitely going to second what Tracy and Jeremy said, but also the fact that water is, and freshwater systems are such a continuous ecosystem. It's really hard to designate specific boundaries between where it affects this part of the ecosystem, but not that part. So tracing sources of pollution and sources of bacterial contamination not only affects that immediate area in the watershed and everything that lives there, but also everything downstream, everything in the ground, everything that's connected to that section of the watershed.
Yeah, for
So the
sure.
connectivity is, I think, what really solidifies it in my mind as an important issue.
Yeah. And then also you as a teacher, you know, we get youth involved, and another good reason to monitor water. Jeremy was just talking about educating yourself so you can educate other people, but also getting youth involved. You know, you're really getting them, you know, future decision makers and voters to learn about what's going on with our local environment as well. So those are all some good reasons why.
And we can also have youth educate elected officials. So one of our groups we're cooperating with is Students Saving Salmon in Edmonds. And they've been collecting a lot of water quality data there and sharing it with their city council. And that's been influential as they try to restore Edmonds Marsh. So, you know, it not only gives kids the scientific experience, but it also gives them experience working with the political system as more than just voters.
Right. Yep, absolutely.
And then I think it also helps to know how some things relate to each other. For example, Eric showed the highly turbid water in his yard and pointed out that not only was there high turbidity, there was also a high bacteria count. And bacteria is not something you can see, but the turbidity is, and realizing that there's a correlation between the two lets people make a real-time decision about whether the water is likely to be safe or not.
Yep. Okay, let's talk about who monitors water. So in our area, there's a lot of individual government agencies that monitor water. In some places where our program is active around the world, there's not a lot of agency monitoring. So here, there's enough monitoring both of our drinking water and our streams that at least from a drinking water standpoint, we don't have a lot of concern. We do still have concerns about water quality issues for habitat and for recreation, but some of the groups that monitor water locally include local city and county agencies, and they may monitor water because they're required to. We'll talk later about some concepts in the bacteria class where water bodies are listed as a known contaminated water body, but sometimes if a water body is known to be contaminated, it's required to be monitored. So agencies may monitor because they have a water quality improvement plan. They're trying to see what's happening with the water. Or they may just monitor for their own scientific purposes for instance, if they're trying to work on restoring habitat or protecting a species of salmon or something like that. But then you also have volunteer groups who monitor, and we have our own reasons for monitoring. Sometimes because not all water bodies are monitored by agencies, or it may be because we want to get a better understanding of the water body, or we want to share that information with others. So our group is called Sno-King Water Watchers, and we're a local program. We operate primarily in King and Snohomish counties, although we've been expanding. We collect data primarily on streams. We do have some river and lake monitoring. We do environmental education, public awareness, and we also investigate and respond to particular problems that get reported to us. We're part of a larger program called Global Water Watch. Global Water Watch is active in a number of different countries. Some countries have got bigger programs, some smaller. There's a pretty large program in Mexico, I just recently was contacted by a program in Chile that's just starting up down in Patagonia. So we're part of this overall network of people as well. And Global Water Watch is, they're headquartered out of Auburn University in Alabama. And they've actually provided us with our training curriculum and our testing methodologies that we're using. So some of the things that we monitor, we do monitoring, which is called biomonitoring, stream biomonitoring. So this is where we're monitoring things called benthic macroinvertebrates, and those are, benthic means they live in basically the bottom area of a stream, and macroinvertebrates are small creatures without internal skeletons. These things can be indicators of water quality because they may be more or less sensitive to water pollution. And so depending on what you find when you do a survey, that can indicate whether the water body is polluted or not. So that's one of the types of monitoring we do. We also do bacteriological monitoring, which we'll talk about later on today. We also do water chemistry monitoring, which involves things like turbidity, temperature, dissolved oxygen, number of other variables. Let's talk about some basic concepts on water and watersheds. One concept is just about how scarce fresh water is and how really scarce fresh water is in lakes and rivers. So if you took all the water on the planet and boiled it down to what percentage of that is actually in fresh water in lakes and rivers, you can see it's a really, really tiny percent of water that's in lakes and rivers. Another key concept is the concept of watersheds. Most of you are probably already familiar with this concept, but it's essentially the idea, it's an area of land that drains to a water body or a stream. And so everything that happens in that watershed affects that water body. So for instance, we're in the Salish watershed, but then within each of the larger watersheds, there are sub-watersheds. So our sub-watershed that we're in right here is the Lake Washington Cedar-Sammamish watershed. And that acronym WRIA 8 is one you may or may not be familiar with, but essentially it stands for Water Resource Inventory Area. The state is divided into different, what they call RIAs, which are essentially major watersheds of the state. So that's our major watershed. All right, we're going to talk about water pollution. Key concept with water pollution is there's different types. There's point source and non-point source, which is pretty straightforward. Point source means it comes from a particular point that you can identify. Non-point source just kind of drains off the landscape. So an example would be you don't have one particular point where this mud is running off, or another example is stormwater running off roads as an example of non-point source pollution. Another type of water pollution, or a type of water pollution, is sediment. So this is an example of a creek that I live on where sediment, basically dirt, was running off of a construction site. And if you see something like this, this is something that you can report. So first of all, you want to identify what it was that you saw. So when and where, what's happening. You're gonna, when you report it to the agency, you're gonna give them your information, and you're gonna notify the local jurisdiction and the Department of Ecology. So in my case, where I've found instances of high turbidity and I've walked back up a creek and traced it to a particular construction site, I was able to report it to, in the case that I found, it was coming from the city of a construction site in the city of Bothell. So I reported it to the city of Bothell and also to the Department of Ecology, and they both followed up to get that construction site to take care of the runoff issue. Another type of water pollution is nutrients. This is a big deal, especially for lakes that can be subject to algae blooms. So phosphates and nitrates are things that would run off of, like, lawn fertilizer is probably a key example of where that stuff comes from. And if you have too much of it, you'll get algae blooms, which can affect oxygen levels and can also be toxic. Feel free to stop as we go along. If anybody has any questions, just ask questions. Another type of water pollution is, as I mentioned, stormwater runoff, toxic substances that run off of the street. There's a lot of different toxic substances in stormwater, including tire particles, chemicals, oils. These have been known to be associated with something called pre-spawn mortality in coho salmon. So I'm going to try and show you guys a little clip on pre-spawn mortality. Oh, you know what? I've found when I do this before.
At first glance, Longfellow Creek doesn't look that bad, but look a little closer.
It's hard to see.
Like these volunteers. Longfellow Creek doesn't look that bad, but look a little closer.
It's hard to see.
Like these volunteers are looking. That brown film could be anything.
Unfortunately, since Longfellow is such an urban creek, the salmon suffer from the toxic cocktail pollutants that wash into Longfellows.
Carrie McGowan coordinates volunteers here, like Emily and Jessica. They're looking for dead salmon. Volunteers are out daily and already got to these.
And probably the same on this one. There. I mean, there's nothing really much to see.
Their bellies are cut open to check for eggs.
From the tip of the mouth.
But first, the salmon are measured.
And so that's about 55 centimeters.
They thought this was a male until the telltale orange spilled out.
Oh, no, it's a female.
It means this salmon died before finishing its life cycle. It's called pre-spawn mortality. And this year, 44% of Longfellow salmon are dying before they spawn.
Which is actually lower than what we've seen in the past for Longfellow. Usually it's around 60 to 90%. And in a natural, pristine stream, it's 1%. It's basically negligible.
Longfellow Creek is a dump site for stormwater runoff in West Seattle. The chemical soup is lethal for coho. It reduces their oxygen level and changes their behavior drastically. Like this one spotted in a near frenzy.
In the past, especially after a rainfall event, we've seen coho swimming in a disoriented pattern, gaping at the mouth, surface swimming. They look like they have no sense of direction, and usually they die within a few hours after seeing that.
It's wiggling.
Sometimes it's even hard to tell if the salmon here are dead or alive.
Do you think that's dead and it's stuck under the trunk?
The Salmon Survey will help conservationists track the health of the habitat here. It also informs plans to reduce flooding and runoff. Longfellow Creek is one of only 2 salmon-bearing streams in Metro Seattle. In West Seattle, I'm Allison Morrow, King 5 News.
Have you guys heard of that pre-spawn mortality? Yeah, unfortunately, something we're dealing with more in the urban area, but that's another reason why we monitor water, I guess, is to look for, basically look for how good the water quality is for what's living in it. What they've found is there's some ways to treat pre-spawn mortality. They've done some experiments at I think it was NOAA Fisheries, where they essentially ran water through just a basic filter, kind of similar to like a rain garden. And they were able to find that the salmon didn't suffer from pre-spawn mortality when they put them in the water that had just been treated with something like that. So there are some ways to work around it.
They're changing the name to urban runoff mortality syndrome because they're finding the same thing happens to juvenile salmon. So coho spend about the first 2 years of their life in fresh water, and they're finding that winter rains or spring rains can wash this toxic storm water into the streams and kill the juveniles as well. So it's a bigger problem than we thought it was originally.
Mm-hmm.
Mostly attributed to a first flush event, or is this more every every heavy rain, spring, winter rain, or?
It's more a first flush problem, but first flush is, even if you dilute it 90%, it's still toxic. So that means that much smaller events can also be toxic to the fish. So, like we just had the rain yesterday that kind of cleaned up our air nicely, but it probably washed a lot of toxins into the creeks. And then, we may have a long enough dry spell that the next rain will also wash significant toxins into the creek. But, there some winters, it rains pretty much every day. And, by the time you're in your 21st day in a row of rain, there may not be a lot of toxins beyond the stuff that's accumulated the day before getting washed in. And depending on the area, you may finally get it low enough that it's not an issue. So they're finding heavily traveled roads like the 520 bridge are highly toxic, and you get into a residential neighborhood where 10 people live there, the runoff tends not to be too bad.
Thank you.
Okay, other types of water pollution can include bacteria. So E. coli in particular is something that can be present, and it can come from human waste, and it can also come from animals. We're going to switch gears slightly and just talk about laws that affect water quality. So in Washington State, the Washington Administrative Code governs water quality, and this particular section talks about freshwater designated uses and criteria, and it regulates temperature, dissolved oxygen, turbidity, pH, fecal coliform levels. There's also other state and local regulations that affect water quality. So there's something called a comprehensive plan that different cities and counties have, and that defines how their critical areas, which include buffers, are handled, and that has a huge effect on water quality. So for instance, if they allow a 25-foot buffer, that's going to have a different effect on water quality than if they require a 250-foot buffer. And that's something that comes up for renewal periodically. So if you follow your local government when they're updating their regulations, that's one area that you can get involved in. There's also other local codes that govern clearing, land clearing and grading, and land development. Periodically, there's an update to a regulatory program called Shoreline Master Programs that regulate waters of the state. So there's a definition of waters of the state, but generally it's larger streams, in some cases large streams, rivers and lakes are considered to be waters of the state. And so that program also governs things like buffers and what development can happen next to creeks. So that also has an effect on water quality. Another area that affects water quality is something called NPDES permits. Sounds very technical. It stands for National Pollutant Discharge Elimination System. And that's something that all cities and counties and some large private developers or private industry has to get a permit with the Department of Ecology that essentially specifies how they're going to handle their stormwater. This is something that gets updated periodically, and this is actually something that has a huge effect on water quality because it really defines how that city or county is going to regulate their stormwater runoff and water quality. Another area of regulations that can affect water quality is tree ordinances. So locally, like for instance in Lake Forest Park, they recently, perhaps a year ago, they revised their tree ordinance, and tree retention and canopy can affect water quality quite a bit. Local decision makers can affect water quality, and in particular, planning commissions usually make land use recommendations, which go to city and county councils. So those can have a big effect on water quality as well. Some things that you can do, and these are just standard things that people are told to, they can do to reduce water pollution. One is building rain gardens, which are things that would collect runoff off of the street, for instance, and instead of just running it into a storm drain where water on the street normally goes into, it would run through this rain garden and get filtered, and it would only run out if the rain garden gets overfilled. So that's one effective way to treat stormwater runoff. Restoring stream buffers is another thing that can.
At first glance, Longfellow Creek doesn't look that bad, but look a little closer.
It's hard to see.
Like these volunteers are.
I don't know why I never restarted. Picking up pet waste, because pet waste, when it runs off, is something that can also go into water bodies and is a known source of bacteriological pollution. Oil leaks, something else that can get into road runoff. Fertilizers and chemicals in people's yards. You know, we talked about nutrients. That's another way that people can reduce water pollution, is not using those things. Green roofs is an idea where people have roofs designed that essentially have soil and plants on them, and that reduces the amount of runoff coming off of roofs. And then pervious or porous pavement is another thing that people can do if they have this installed in their yard. It can help reduce stormwater runoff into creeks. Okay, talking about water monitoring, the steps of water monitoring are, first of all, you make a plan. So what are you going to do? Where are you going to do it? Why are you going to do it? You pick a site. We'll cover each of these things in sequence in a little bit more detail. We'll talk about how you record the data once you start water monitoring, how you can present and interpret the data. So the steps, first of all, you're going to define your goals. So why are you doing it? You start gathering information about the watershed that you're going to be monitoring. One thing that you want to do is learn where that stream goes within the watershed. So you're going to do something called a stream walk. You want to identify where are some areas that either are especially important, like something is affecting the creek, or it's a place where you can access the creek, maybe a strategic site for monitoring. We'll give you some criteria for selecting sites that you might want to monitor at. And then you start collecting data and put your data to use. So those are the basic steps of water monitoring. So we talk about picking a water monitoring site with C.L.A.S.S, which stands for convenient, legal, accessible, safe, and strategic. So when you want to choose a site, you want to make sure that, first of all, it's close, like convenient example, it would be close to your house as opposed to far away. Legal, you make sure that you actually have permission to get there. Accessible, you can actually make it to the site. Safe, there are safety factors, parking, streets. You want to be in a place that maybe has got some visibility, so you're not going to an isolated place by yourself. And then strategic, make sure that you're learning what you want to learn from that site. So with our program, there's certain ways that we record the data. So we have certain forms and certain ways that we submit the data. So initially, to establish a site, we have a monitoring site form. And this is in page 33 of the, I think, the introduction to water monitoring manual that I emailed you guys. So this just defines essentially who's going to be monitoring there, where it is, some details about it, and this is what Global Water Watch will use to set the site up in its database. Then we have different reporting forms for different types of report monitoring. So if we're doing physical or chemical or bacteriological reporting, we have particular data forms. This is an example of the physical and chemical monitoring data form. So you've selected your site, you started water monitoring, you've collected your data, you entered it on the form, then you're going to enter it into a Global Water Watch online database, which tracks monitors, it tracks locations, it tracks the water quality variables, And then it gives you the ability to view that data with different maps and graphs and tables. On the Sno-King Watershed Council website, we have a link to a data entry and viewing page for Global Water Watch. So on that page, it takes you to a place where you can sign in to Global Water Watch. And then here you log in, and once you've logged in, you can enter different types of data. So I can either view data or I can enter data from this point. This would be the data entry screen under, once I said I'm going to enter a particular type of data, and this is water chemistry. So this form would pop up. You just tab through and enter the data in this form, and it goes into the Global Water Watch database. So now you've gone through all that process. Your data is in the database. Now what are you going to do with it? So there's different ways that you can share your data with others. One thing you can do is you can look at it graphically, and you can compare data from different sites. So this is an example from Alabama, where they monitored a variable called alkalinity. They found a super high alkalinity at one particular point and found that by comparing the level at different sites, they were able to identify it was coming from an industrial source. So that's one thing you can do with your data. Another thing you can do is you can look at the graphs and see, okay, maybe you have a high level. This is an example of a high level of bacteria that was found on the creek that I live on, but higher upstream. You remember we were talking about, 100 being safe level in Washington State, 200 is the Global Water Watch standard. And you can see that we're approaching 20,000. So we took this data that we collected and notified the city of Bothell, and they did some investigation to try and help us figure out where is this high level of bacteria coming from. You can also use report cards. So one of the things we worked on is producing report cards for our different sites. And so that's something that you can share with people and give them some information about what's going on with your creek and your site. This is the Student Saving Salmon program that are talking to the City Council in Edmonds. So that's another thing that you can do, is you can share your information. So your next steps in terms of water monitoring would be learning about the stream you want to monitor and its watershed. Look at things that might affect that stream. Think about your monitoring goals, make a plan, become a monitor with us, or maybe with another group, like if you're already associated with, for instance, Lake Forest Park Stream Keepers, and then put your data to action. So I kind of blasted through a lot of information in a short period there, but do you guys have any questions on that stuff right now? That's just kind of a general overview of introduction to water monitoring.
I have a quick one, Eric. On the data, once it's entered into the database, can you then re-export it if you want to run it through a different, say, visualization software or something like that? You want to do something else with it?
You can. And right now from Global Water Watch, you can either export it to an Excel spreadsheet directly out of their database on a site-by-site basis. We can also make requests to them to produce a larger export of data. We're also right now working with a group called Water Reporter that is going to allow us to, on more of a large scale, export data from Global Water Watch's database and display it graphically in a map on our website.
Thank you. Great.
Sure. Okay, well then I think what we'll do, if you guys don't have any more questions, we'll jump right into bacteriological monitoring.
Eric, could I ask a quick question?
Yeah.
On the, in your example on Little Swamp Creek, you had a specific event that you were concerned about. Did you go out and do additional sampling as a volunteer, or was it more of a, you saw this happening and you made the call? Because we're typically monitoring the same day, same time of day even. to get a baseline. Did you jump out and do extra stuff that day?
When I saw that high level of bacteria,
yeah, or that turbidity, that one. I don't know if that's the same example. So.
The turbidity in particular is something that you can take action on. There's something usually when you see something like that, it means that it's more than just like a normal storm event. It means that you have probably like a construction site that is not adequately controlling its runoff. So when a new, like say a new housing development in the area I live in, there's a bunch of new housing developments going in, and they're supposed to have what they call temporary erosion controls, temporary erosion and sedimentation controls, TESC measures in place. And some of them are really great about that. Like if you walk around, you'll notice that some construction sites, they'll have like a big pile of dirt and it's covered with a tarp, and they have a bunch of different sort of mulch on the ground. And other ones you'll see, they just have a bunch of dirt out there and it's not even covered. So some cases, the ones that aren't so great, they just kind of maybe hope it doesn't rain a lot. But if it starts running off, that's something that they're supposed to control. I mean, it's regulated like an oil spill. So that's something where, like the example I gave, where I'm talking to, or I reported it to the city of Bothell and reported it to the Department of Ecology, they went out and took action and actually made the construction site stop work until they had corrected their erosion problem. So some of those things, if you find something, you can take action on it right away, and that's a good example of it.
Gotcha. Thank you.
Sure. Okay, so we're going to talk about bacteriological monitoring. Already kind of talked about our organization and about Global Water Watch that we're a part of. This is an example of a bacteriological monitoring workshop that Global Water Watch was doing in Kenya. We've already covered why is it important to monitor water. A couple of the good reasons are, is it good habitat for wildlife, and is it safe for people? And finally, is it getting better or worse, and why? So we know that water quality is important for salmon and other things that live in the creeks, and things that depend on those salmon. Water quality is also important for people. We're lucky that our drinking water quality around here is good, but from a recreational standpoint, a lot of times people are not aware of the impact that water quality could have. And so in particular, bacteriological water contamination is something that people don't really think about, but people recreationally access little streams, like kids go play in the streams, where the stream may actually not necessarily be safe for them to swim in or play in. This is an example again of Little Swamp Creek when the water quality wasn't so great and we had the high bacteria level. Let's talk about bacteria. They're single-celled organisms. You can't see them. They're all over the place. They're on us. They're inside of us. They're necessary to a lot of processes in life. Essentially, they help break things down and transform things from one form of matter to another. And so they're integral to life. They come in different families. So the ones that we're mostly concerned with are a type of coliform bacteria, and in particular E. coli. But you'll see that the sampling that we do is for, in particular, it tests for E. coli and other coliforms, and the color coding on that tree sort of represents what you'll see with the color coding, the color that the colonies show up in our bacteria tests. So E. coli is the thing that we're testing for. E. coli is indicative of fecal contamination, but it isn't necessarily 100% certain that it means fecal contamination. So when we see high E. coli, it indicates likely high fecal contamination, but it isn't necessarily synonymous with that. around the world, this kind of contamination of water is a major problem, and which is why a lot of countries around the world have unsafe drinking water. You know, in countries like Haiti, where I've done some volunteer work, they don't have sewage treatment in a lot of areas. And so, for instance, they were dealing with a big cholera outbreak there. The main thing that we're checking for around here is a particular variety of E. coli, and that's the variety that can make you sick. Here's an example of where E. coli was traced to a water park, and I think I've got a little video.
A popular body of water in Shelby County is filled with dirty water.
The Cahaba Riverkeeper recently discovered high levels of E. coli in Buck Creek. Tonight, ABC 3340's Honora Gathings is fighting for your health. She is live in Helena at Buck Creek. And Honora, is it safe to swim there?
Well, Dave and Brenda, the executive director of the Cahaba Riverkeeper, says a dip in this water could make you sick. There are no federal or state agencies regulating Alabama's creeks, and certainly no signs warning about dirty water before you take the plunge.
Janet Hernandez and her 4-year-old son often skip rocks in Buck Creek, but they have a rule: feet and only feet first.
It's just our feet. I would freak a bit more if like he started swimming in it.
Other families don't hesitate to wade on in for fishing, swimming, or tubing, but Hernandez,
To swim in it, I don't trust it enough.
less than a foot from the family, a layer of gunk floated near some rocks. We sent the executive director of the Cahaba Riverkeeper a picture. Dr. Meyer Crawford believes it's either algae or scum. She says neither one is good.
Very few people are doing any kind of testing.
The Cahaba Riverkeeper started testing 8 points along the Cahaba River last summer, starting at Buck Creek and ending at Trustville Springs.
No one else is doing it. I think it's pretty important.
The results were documented and photographed. The dark spots are E. coli. Crawford says unsafe levels were found in Buck Creek multiple times, but an April 14th sample set a new high.
It's called too numerous to count. You can't even count the number of little dots.
Crawford says any E. coli can make you sick.
You don't even have to drink it if you have an open cut. If you're just wading and you have an open cut on your leg, or you could become infected that way too.
E. coli comes from feces, but no one is certain what is polluting Buck Creek. The EPA is monitoring 3 companies upstream of Helena for toxic releases and more than 100 others for water discharge. But there are no current violations. The Alabama Department of Environmental Management says it isn't aware of any either. Helena's wastewater treatment plant is just over the Buck Creek Dam, about 1/4 of a mile upstream, but it is also in compliance and closely monitoring its discharge.
We're going to find out, I hope.
But until then,
I'd rather take the kids to the pool or something where it's really clean. They can actually swim.
Now this is a look at the substance that we sent a picture of to the Cahaba Riverkeeper. In less than a month, the Riverkeeper will resume testing of this water. The focus, though, will be over this dam upstream to help identify the source of the E. coli. The Riverkeeper will post the results to his Facebook page every Friday. Fighting for you in Helena, Honora Gathings, ABC 3340.
And if
So I thought I'd just check in and see if you guys have any reactions to that or any thoughts about that contamination issue. Something that we have around here as well. So when you learn about, we'll talk about some of the things that creeks are so-called listed for, but creeks can be listed for different pollutants, for having known too high levels of different pollutants, and fecal coliform is actually a pretty common one around here. So for instance, Swamp Creek, we're in the Swamp Creek watershed, has a plan and a listing for too high of level of that. So it's pretty common around here, unfortunately. So where does it come from? We talked about pets is one place where it can come from. This is a relatively recent article last year, I guess, in Kirkland. Kirkland did an interesting study. So they were, this was, I think, at Juanita Beach Park. They did a study where they had volunteers go around and plant little flags everywhere that they saw dog poop. And they were incentivizing people to pick up dog poop. And they were also documenting how much dog poop there was. And so basically what they found was two things. One, there was a huge amount of dog poop being deposited at this park. And two, by pointing it out with these flags and talking to people about it, they were actually able to get people to change their behavior and reduce the amount of dog poop that was being left in the park. Another place that bacteria can come from into streams and water bodies is from these things called side sewers. So people's homes are connected to the main sewer line through this side sewer. And these things can occasionally leak or actually get improperly connected. One of the groups that we were working with is called the Thornton Creek Alliance, and Thornton Creek has very high levels of bacteria. It's one of the major watersheds in Seattle. And so Thornton Creek Alliance did a citizen science project in conjunction with Seattle Public Utilities, where they went out essentially weekly with volunteers and monitored multiple sites throughout the watershed for bacteriological contamination. And ultimately they found, when they compared the results all around the watershed, they found some areas that were really high. And when they did some more, they narrowed that testing in, they found essentially a apartment complex that had an improperly plumbed sewer line that had been plumbed into the storm sewer. So it was basically putting sewage into the creek. So that wasn't all of it, but that was one of the major ones. So that's a place where things can come from either improperly, or if these just fail on their way to the main sewer line, they can just start leaking into groundwater, which can make its way to the creek. Animals, livestock in particular, is another thing that can contribute to water pollution. Also birds. Here's an example from the University of Washington, Bothell, where they have a gigantic crow rookery, and tens of thousands of crows nest there every night. And not surprisingly, where North Creek runs through the UW Bothell, it picks up a lot of bacteria as well. Let's talk about how we measure bacteria. This is an example of the equipment that's being used to monitor the bacteria. So they have a few basic principles, Global Water Watch does. One is do it the same way, same place, same time. They advocate using this simple low-cost method. If you do it the same way using consistent methods and techniques, you can generate credible data. And always, if in doubt, do it again. So in other words, make sure you don't make a mistake when you're doing the technique. If you do it over, so you're not generating incorrect data. So we're monitoring for E. coli and general coliform bacteria. And these are examples of what we're going to be using for our monitoring. When we talk about how we measure bacteria, we measure it in terms of things called colony forming units that are abbreviated CFUs. So this is an example of an incubated petri dish of water and media where the E. coli colonies react with an enzyme and turn blue, and other coliform bacteria's colonies result in a pink color. And so what you do is, we'll talk about how you count them, but essentially you identify the number of those dots that are big enough for you to count and come up with a number of colony forming units. So again, a CFU is a measure of a colony of those cells that's viable enough to form a colony. So in other words, if you just had a tiny dot, you probably wouldn't even see it. But if it's big enough to grow into one of these larger colonies after you incubate it, then you would count that as a colony forming unit. The standard that agencies and people use when they're talking is the amount of CFUs per 100 milliliters of water in your sample. Global Water Watch has some standards that they train us on for what is a risky level or what is not a risky level. So in the Global Water Watch standards, greater than 600 CFU per 100 milliliters is considered unsafe for contact. 200 to 600 is safe for infrequent contact, and less than 200 is generally considered to be safe. Washington State has stricter standards, and so they changed their standards recently and simplified them somewhat, but essentially they came up with less than 100 is considered to be safe, greater than 100 is considered to be risky. We talked about a concept of what's called listing. So a couple concepts here. One is what's called a 303D list. Section 303D is a section of the Federal Clean Water Act. And under that section, a creek or a water body can be identified as being impaired due to a specific pollutant. And that could be bacteria, but it could also be low dissolved oxygen. It could be high temperature. So if somebody collects data, somebody thinks that a creek might have a pollution problem, they collect data that says, okay, the bacteria level is too high or the temperature level is too high, then they'll submit that data to the Department of Ecology, who add it to this 303D list. So if you want to look up which water bodies are polluted with different types of pollutants, you can search for 303D listings for the creek that you're concerned about. Another concept is a thing called a water quality improvement plan. So for instance, let's take the example of Swamp Creek has a listing, a 303D listing for fecal coliform bacteria. So the city of Kenmore and other agencies that have jurisdiction over Swamp Creek have to have a water quality improvement plan where they say, we're going to put these measures in place to improve the water quality of Swamp Creek. That brings into play another concept, which is the total maximum daily load, which is referred to by the acronym TMDL. So that's essentially what is the maximum level of pollution we can find in this creek. And they're hoping that it will be less than that. But essentially they're saying, we're going to monitor this water body monthly and see how the level of pollution that we find for this variable compares to what the criteria is in the TMDL. So a creek gets listed on a 303D list. They develop a water quality improvement plan that establishes permissible levels for pollutants. So a little bit technical there, but that's just to give you some vocabulary about water pollution and bacteria. Okay, so how do we actually do the monitoring? So we have some different materials that we're going to use. We're going to use Petri dishes. We're going to use this stuff called Coliscan Easy Gel, which is a medium that essentially it's like food for the bacteria. We're going to use a pipette to collect our sample. We're going to record the air and water temperature with a thermometer, and we'll also record a temperature while it's being incubated. We're going to use an incubator, and there's different designs of incubators that you can use. Global Water Watch teaches building a simple low-cost incubator using essentially a cooler, like a nightlight, and a little cord. We originally started with that model, but we've ultimately moved to using an egg incubator with a thermostat because it's just easier to work with. So you're going to go out and go monitoring. You're going to get your other materials together. You need your data sheet. Once you collect your sample, you can either transport it with not on ice less than 30 minutes before you incubate it, before you plate it and incubate it, or if it's going to be greater than 30 minutes, you need to transport it on ice. So we bring a little ice pack with us in a container. We're going to get our Petri dishes ready, and you'll learn there's a certain way that we basically put tape on them so that you can open it up and then seal it up so that you can put it in the incubator and not get any additional contaminants in it. So we'll pre-label our Petri dishes and our media before we do our sampling. And you can see that there shows 3 different samples. So for each site that we're going to sample, we're going to collect 3 what we call replicates. So 3 different samples, and then those get averaged to come up with your number. So we'll label them to make sure that we're not accidentally putting our 2 samples in the same dish. So we're going to have, for instance, 3 samples here, 3 Petri dishes and 3 samples that correspond with that. So in terms of collecting samples, when you go out to the site, you're going to unwrap a sterile pipette, collect the sample. You want to be facing upstream so that you're not interfering with the water that you're sampling. And you want to be below the surface of the water, but not on touching the bottom. You'll collect the sample and put it into the media bottle and then recap it. And then you bring it back for plating. And I'm going to show you guys a video on how to do this.
Welcome to the Alabama Water Watch instructional video series. This video will show the proper method for collecting water samples used to determine the number of bacteria in the water body. This is the first step in a 3-step process. The second and third steps, plating the samples and determining results, are covered in their own separate videos. For each site being tested, you will need a cooler with ice, 3 media bottles at air temperature, a sterile pipette, and a thermometer. Label your media bottles by site and replicate. If you sample multiple sites, be sure to use separate data sheets or keep record of monitoring site and bottle numbers that are grouped in sets of 3 replicates. For example, if you have 2 sites, you can number the replicates at the first site as 1A, 1B, and 1C, and the replicates at the second site as 2A, 2B, and 2C. Upon arrival at the site, measure the air and water temperatures. For instruction on how to measure these temperatures, please watch the video called Water Chemistry, Air and Water Temperature. Once you are ready to sample, unwrap a sterile pipette from the bulb end and avoid touching the tip of the pipette with anything except the sample water. Fill the pipette with sample water. Be careful to not disturb the bottom of the stream. Carefully squeeze the bulb and expel excess water to the desired sample volume. 1 milliliter sample volume is recommended and is usually sufficient. Be sure to use the same volume for each of the 3 replicates and record volumes on data forms. Remove the cap from the media bottle and hold it without touching the inside. Squirt the sample water into the bottle of liquid media without placing your hand or face over the open bottle. Cap the bottle securely and place inside the cooler. Repeat these steps until all 3 media bottles are inoculated with water from the same spot. Keep in mind that media must be plated within 3 hours of collecting the sample and should remain on ice and out of direct sunlight at all times. Thanks for watching. If you have any comments or questions, give us a shout.
All right, you guys have any questions so far? No? All right. Dave, you got anything to add?
I've got a question, Eric.
Sure.
Where do you get this stuff? Is there like a kit you have to buy, or do you just go out and get it on your own?
So if you're monitoring in a program that's associated with us, then we'll provide you with the supplies. Yeah, I didn't mention that in the very beginning, but we are operating under a grant right now from King County Waterworks program. And so that is paying for our water monitoring supplies, like our bacteria monitoring supplies.
Cool.
Okay, so that was collecting the sample. So the next part that you do is what's called plating the sample. So we've gone to the stream, we've collected our sample, we put it in the bottle. One thing we didn't mention, they talked about the sample size. We'll get into sample math a little bit in the future, or in the next couple slides. But so we bring our sample back. Now we're going to do what's called plating. So we're essentially going to pour the sample into the Petri dish, and we're kind of moving to a slight change from what they used to do. We used to plate the samples, let it gel, because they're pre-treated with something that will cause the sample to gel up, then put it in the incubator. Now we're essentially just pouring it, plating it directly in the incubator. So we do this plating process with the dish in the incubator. So you'll open it up, pour the media in, swirl it to cover the bottom, but not too much because you don't want it going way up the sides. That'll make it difficult to count colonies. Like I said, we used to let it gel first. Now we just plate it right in the incubator and then incubate it. So I'm gonna show you guys a video on how to do plating. Oops, not that one. Plating.
Welcome to the Alabama Water Watch instructional video series. This video will show the proper method for plating water samples used to determine the number of bacterial colonies in a water body. This is the second step in a three-step process. The first and third steps, collecting the samples and determining results, are covered in their own separate videos. Prepare your Petri dishes by taping the top, the larger piece, to the bottom, the smaller piece, on one side of the dish. Keep the dish closed as much as possible to avoid contamination. Label the plate lids with labels that correspond to the inoculated media bottles in data form. Lift the top of the Petri dish just enough to pour the contents of its corresponding media bottle into the plate. Do not slosh the media onto the sides or lid of the plate. Record the time you plated your sample on your data form next to incubation start time. Be sure to shield the sample plates from direct light. Leave the plates undisturbed until the media has gelled, usually about 1 to 1.5 hours. After the media has gelled, use another piece of tape to secure the untaped side of the dish. To prevent condensation from dripping onto the plates from the lids, turn the plates over and place them in the incubator. Incubate plates between 29 and 37 degrees Celsius for 30 to 48 hours. Thanks for watching. If you have any comments or questions, give us a shout.
Okay, any questions on plating?
So I assume if we're placing them directly in the incubator rather than letting them gel, we're skipping the flipping part?
Yeah. Yep.
Right.
Yeah, it's just, it has to do with somehow over time the Petri dish production has changed or something so that they weren't gelling up as well. And maybe it has to do with people using larger samples, because you can use anywhere from a 1 to a 5 milliliter sample. Typically we use like anywhere from about 1 to 3 milliliters, but if you have a larger sample, it takes longer to gel. But it just proved to be kind of a unnecessary step, and sometimes it didn't gel in the exact amount of time. And so it just ultimately Global Water Watch issued a update to their procedures, and said go ahead and plate it directly in the incubator.
And do we have more specific, well, two questions. First of all, do we make our own incubators, or do you provide them?
So we provide the incubators, and actually, hang on a second, I got one right here. So the incubator that we use looks like this. So it's got it basically is this stuff taped on it it comes with Windows. It's an egg incubator, but basically it's got a thermostat. And so we just plug this thing in, open it up, and put our dishes inside of it. And then what's nice about this is you can program in, and we pre-program these with the set temperature. So it maintains the temperature like it's supposed to for the whole time. With our homemade incubators, we used to have an issue with, if one person would, if we transferred it from person to person, one person would put it in the garage, another person put it inside their house, and we might get our temperatures out of range. And so just it lets our technique be more consistent.
And the incubation time is fairly broad, between 30 and 48 hours. Are we more specific about that?
No, essentially you just have to give enough time for the colonies to grow. I think if you go beyond that, you might get like a lot colony overgrowth, or it might be hard to count the separate colonies distinctly. So,
okay,
yeah, and that gives you a window depending on when you do your plating. If you do it it just lets you set the clock. So if you do it in the afternoon, you wouldn't necessarily have to then do your counting in the middle of the night, if it timed out a certain way.
Fair. Yeah. Thanks.
Okay.
Yeah. And actually, if you do run it a lot longer, it's usually fine. So I think what happens is the medium dries out, and once it's dry, things stop growing. So you really don't have that overgrowth problem unless you like leave it in a bathroom where people are taking showers all the time and the humidity is really high. And then I guess the other thing that can happen if you let it run a little long is you start getting other things like mold growing on there that can obscure things a bit. But I've had some that I've run for a month, and basically look the same after a month as they do after 30 hours. So running too long doesn't make a difference, and then running too short, the colonies are smaller. So we have a size criterion for what's big enough to count and what isn't. And some of the ones that would grow to be big enough to count aren't there by 30 hours, but by the time you get to 40, they're clearly big enough to count.
Dave, did you intentionally let those go for a month, or was that an accident?
It's intentional, and I've got some that instead of killing them the way we're supposed to, I've let them continue to run at room temperature for over a year, and they still aren't changing very much.
All right. Well, we're not advocating that.
The correct procedure is to kill it with a spoon of bleach after you've counted it, and that way you don't have to worry about infecting yourself if you got a dangerous strain that you happen to colonize. But I'm a biologist, so I have different curiosities than other people might have. And I've also been known to let whales collect my samples for me. So they exhale and blow a bunch of water into the air, and then I'll collect the droplets as they settle down and culture those.
Okay, so the next step, so we collected our samples. we plated and incubated, then our next step is going to be, we're going to determine the results. So I've got another little video to show you guys on how to do that.
Welcome to the Alabama Water Watch instructional video series. This video will show the proper method for determining the results of incubated water samples. This is the third step in a three-step process. The first and second steps, collecting the samples and plating the samples, are covered in their own separate videos. Remove plates from the incubator, being careful not to spill any liquid. Be sure to record the incubation period, which is optimally between 30 and 34 hours. When counting the number of colonies, it is helpful to use a white sheet of paper to split the plates into quadrants. Count all blue to purple colonies on each plate as E. coli. Do not count very small or pinpoint colonies smaller than the period at the end of a sentence. Count or estimate the number of all pink to dark red colonies on each plate as other coliforms. Estimates of more than 100 colonies should be reported as abundant, and estimates of more than 200 colonies should be reported as too numerous to count. This sample should be recorded as too numerous to count. If colonies appear as a ring around the edge of the plate or as one large blob, count as one colony. Record both the number of E. coli and other coliforms on the data form. The bacterial concentration in 100 milliliters will automatically be calculated in the Alabama Water Watch database. To calculate the bacterial concentration by hand, divide the number of E. coli colonies counted by the sample volume, then multiply by 100. Kill the bacteria by pouring about a teaspoon of undiluted bleach into the dish. Let stand for about 15 minutes, then dispose of with household garbage. Thanks for watching. If you have any comments or questions, give us a shout. Welcome to the Alabama Water Watch instructional video series.
So just to clarify a couple things on there. One is they said 30 to 34 hours, so that was just a mistake. And then as far as cleanup, Dave was talking about the bleach. I've had some other people ask me if it's just okay, if you're not going to reuse the Petri dishes, can you just put them in a Ziploc bag and dispose of it, landfill it? And I don't really see a problem with that. That's not the official technique which is taught, but I think that you could also dispose of it that way. But the way that they teach to do it is to open it back up, pour some bleach in it, let that sit in there for a little period of time, and then you can basically dispose that, or you can dump that. It'll slide right out. You can just dump that down the toilet and get rid of it that way. I don't know, Dave, do you have anything you want to add about that?
Yeah, if you don't kill it first, then you run the risk that something is going to eat it. And again, if you have a dangerous strain, which is rare in E. coli, but it does happen, a mouse or a rat or something is going to get a very large dose that it can start dropping in the environment for other critters to pick up, or to wash into a stream where people can pick it up. So it's probably best to kill it. But as I said, I've let some of my colonies run over a year and I haven't gotten sick from it. So it's sort of a low risk thing if you don't bother to kill it.
All right, so we're going to talk about how to count bacteria. So we've done our incubation, we pulled it out of the incubator, and now we're counting. So how many, let me know, you guys, how many E. coli colonies do you see in there? So those would be the blue colored ones.
Looks like 7.
Yeah, I confirm.
Also what I see.
Okay. So 7. So then how many, so our sample volume was 1 milliliter. So if there's 7 in 1 milliliter, what's the number in 100 milliliter?
What is 700?
Sounds like a Jeopardy. So there's the math, and correct, 700. So then the number of other coliform really depends on if you can count them or not. And another thing that you can do is you can see that this has been divided up into 4. You can divide, I essentially take a Petri dish lid and I've made it a grid into 8. So I could divide it into 8. So I could count the amount of other coliform that are in 1/8 and then multiply it out. So you go through and count all those colonies. You don't count the ones that are super small. So in this case, it counted 45 and 1/8. So our other coliform is 360, which would be 36,000 in 100 milliliters. We don't really worry about that number. That's kind of just a reference number. We record it, but the E. coli is really the key number that we're concerned about. The other number would be more like, it'd be unusual if you found no other coliform when you had a bunch of E. coli. So that might tell you there was something going on with your sample or with your materials. Going back just so to interpret this, so would 700 milliliters or 700 CFU in 100 milliliters, 700 colony forming units, would that be a good source for drinking water? You guys remember what the level is?
I sure hope not.
Yeah, so our cutoff would be 100. And really, I mean, even 100, I don't know if you'd want to drink it, nor would you want to swim in it. So here's the standards for Global Water Watch. Washington state standards are even stricter still. So here's another example. So this is kind of like a math example. So in this case, our sample volume was 5 milliliters. And how many colonies do we have here?
7 still.
Yeah, it looks like 7. So if we had 7, our formula is the number of colonies divided by the volume of our sample times 100. So our number of E. coli would be 140 CFU. So how does that compare to our standards? Do you guys remember?
Still wouldn't drink it.
Yeah. Still wouldn't drink it. By Washington state standards, you really wouldn't want to be in it, but it's less risky. It's not super risky.
Right.
Yeah, so I'd probably canoe or kayak in 140, but I wouldn't swim in it and definitely would not drink it.
Yeah. So these are just some other exercises we could do with the math, but essentially you guys get the idea of if you vary the sample size, you have to adjust your math to come up with the number of colony forming units per 100 milliliters. So once we've done our counts, then we record it on this data sheet. And so you can see all the different things that we record on the data sheet, the information about the monitor or the site, the water body conditions, air and water temperature, what was our sample volume, how long did we incubate it. The incubation time is what time did you start incubating it? And we put that down as the time when you plate the sample as your incubation time. And what was the temperature, how long? The media does expire, so we always check before we monitor to make sure it hasn't expired, and we record the expiration date on the form. Plate it on site. We generally don't plate it on site, and you may or may not transport on ice. And then there's a place to record the E. coli and the other coliform. So then you do cleanup, which we kind of talked about. And then ultimately you're gonna do something with your data. So you can kind of interpret, think about what you're finding and what does it mean. So if you're finding it at a place that's commonly used for recreation, maybe you wanna share some information with, if it's a public site, maybe you wanna find out what agency controls that site and say, hey, maybe you guys could post a warning sign, or would you like us to post a sign? Or you may find that, there's a high level, like for instance, with the Thornton Creek example, they looked at multiple sites within a watershed. They were trying to pinpoint a source. And so they used that to find their source. And they zeroed in after monitoring multiple sites to monitoring in a particular, closer to where they were finding the high levels. And so you can use, this is an example of the graph off of the Global Water Watch site. So you can use this to present data. You can also turn it into a poster. So here's something that Lake Forest Park Streamkeepers came up with, showing kind of what it normally looks like when they sample, and then what it looks like after a heavy rain. And so that's a great graphic example. And I think they used this when they were having some of the, I think they called them Green Days events at the town center in Lake Forest Park. You can do something like what I talked about with the Thornton Creek example, where you can collect data simultaneously if you're trying to pinpoint a source within a watershed or a particular stream. Here's an example where at Auburn University, they found super high levels of contamination at a pond in a thing called the Memory Garden. And so they put up a sign warning people about it. So there's different things that you can do with your data. Here's an example of the Global Water Watch program working in Kenya. They had people take the samples and they drew a watershed map on the floor. They laid out all their samples, and then they transferred that into a larger scale watershed map so they could identify where they had water quality problems and where they didn't with regard to bacteria. Another way that this is used, there's some groups called waterkeeper groups, and they have a thing called the swim guide, which they identify bacteria levels at swimming beaches. And so this is another thing that can be done. In some cases, oftentimes swimming beaches like around here are monitored by a health department, but there are instances where a volunteer group might do that monitoring, and the swim guide is an example of that. So this would be off the swim guide. They could say, okay, is this a place where I typically want to go swimming or not? Here's an example from Global Water Watch Mexico, where they just had an informational poster they put showing, comparing different sites and showing what the results were in the different sites. All right, and these are the folks that helped us get our program going and have been funding our program. So that is pretty much it for the classroom portion of this. What I'll do is I'll email you guys, or either email you in advance a registration form, or bring it when we get together for a practical session. Now, are you guys going to do water chemistry next week as well? Okay, and then Jeremy, you've already done water chemistry, but do you want to go through it again since it's been a while?
Yeah, I could. In addition to COVID, I had a kid in between being trained, so that's the other reason I've been out much.
Okay.
So yeah, is that next Saturday?
Yeah. So what we'll do is we'll do that classroom session, and then after that session, I'll schedule a time that either we can all get together as a small group, or if we're not able to get together as a small group, then we can get together individually and just make sure that you guys have a chance to practice the hands-on skills, and I can verify that you've got the hands-on skills, and then we'll submit your information to Global Water Watch and get you set up as a monitor, assuming that's what you want to do. So cool. All right. Any questions at this point?
I have a question. How long does the grant run for?
Our grant is a 2-year grant that is going to cover us. Well, it's probably going to get extended. Our grant is going to cover us at least through the end of 2021 and probably through 2022.
Okay, good.
Yeah, and so far we've been successful. We had an initial grant from the Rose Foundation and also from Fish and Wildlife Aquatic Lands Enhancement Account, and then we've had a couple rounds of we've had funding from King County Council member grant Rod Dembowski, and then a couple rounds through King County Waterworks. So far we've been lucky at keeping our program funded.
Mm-hmm. If I'm not being too nosy here, about how much does it cost to run something like this?
It's not super expensive. I could give you the whole program budget, but I think our grant for 2 years, and that includes some other types of testing, is about $25,000.
Oh, okay.
Yeah, and that actually includes a whole nother section of testing for BIBI. So really basically the cost for monitoring for all the bacteria, just for the ongoing cost of monitoring, is about $10 per site visit.
Oh.
And then you extrapolate that out times number of sites and frequency.
That is thank you.
Yeah, I think the chemistry, we go through somewhere between $1 and $1.50 worth of supplies per site. So again, that's something where you do a site once a month and then multiply by the number of sites and months in a year to get the annual cost. Since we've got volunteers doing it, we don't have labor costs to worry about, which for professionals is a big part of the budget. And a lot of the countries doing this are, what we think of as third world countries, where people are likely to have to pay for it out of their own pockets, which are not likely to be very deep. So folks at Auburn University spend a lot of time trying to figure out what inexpensive ways are available to produce reliable results. And, this is what they've come up with. And, sort of the incubator design they came up with, costs a few dollars to put together, and for a few dollars more you can add like an oven thermostat that'll keep the temperature consistent within a couple degrees, and then if you're in better shape financially, you can go to these egg incubators that are a little less than $100, but they'll keep the temperature consistent within, say, 1/10 of a degree. But the results you get are pretty much the same whether you let the temperature fluctuate a couple degrees or 1/10 of a degree, or you get a university grade one that'll keep the temperature constant, say within 1/100th of a degree. It turns out that on the scale of a colony, there's not a lot of difference over that 29 to 37 degree temperature range, and there's not a lot of variation between 30 and 48 hours. So at the level we're trying to do things, it doesn't take a big budget.
Okay.
All right. Well, thanks, you guys. So I'll sign off for now if nobody else has any questions, and I'll see you guys next Saturday.
Right.
Thank you.
All right.
Thank you, Eric. See you
Bye.
next time.
And Eric, can I ask you a question offline in a minute?